does not Lyophilization does not begin in the freeze dryer. Wait a second. How can the freeze drying process not start in the freeze dryer? That just doesn't sound right, does it? This one sentence basically challenges everything you'd assume about how this works. Exactly. That's the big question we're tackling in this explainer. If not in the big, fancy machine, where on earth does this super critical, super precise process actually kick off? You might be surprised by the answer. Believe it or not, it begins right here, with the packaging. Yeah, the simple glass vial and its stopper. It's not an afterthought, it's not just a box to put things in. It is the actual starting line for the entire lyophilization process. And this is the main idea we really got to wrap our heads around. We have to stop thinking of vials and stoppers as just, passive shells made of glass and rubber. In freeze drying, they are active, functional parts of the system, almost like components of the machine itself. They directly shape the final outcome. So whenever we say packaging from now on, this is what we're talking about. The primary packaging. Anything that physically touches the drug product is part of this critical system. It's the medicine's immediate personal environment. And why does this matter so much? Well, the stakes are incredibly high. For today's most advanced medicines, I'm talking about things like biologics or the mRNA vaccines we've all heard so much about, getting the packaging right isn't just a small detail. It can literally be the deciding factor between a stable, life-saving drug and a completely useless vial of liquid. And just so we're all on the same page, biologics are extremely sensitive molecules. They come from living systems, which makes them really powerful, but also really fragile. They need a perfect, stable environment to work. And guess what? That perfect environment starts with their packaging. So, how does this all actually play out? Let's get into the process itself. We're going to walk through the 3 critical stages of lyophilization, and you'll see exactly how the packaging plays a starring role in every single one. Okay, here's our roadmap. It's a 3-step dance: freezing, then primary drying, and finally, secondary drying. And for each step, you're going to see the vial and the stopper are right there in the spotlight. First up, the freezing stage. This is where the liquid drug gets carefully frozen solid. And it's not just about making it cold, it's about forming a very specific ice crystal structure. That structure is absolutely essential for what comes next. Now check this out. The vial isn't just holding the liquid, it is controlling how it freezes. The thickness of the glass, that determines how fast heat can be pulled out. And even the physical shape of the vial's bottom, the geometry, can dictate where and how those first crucial ice crystals start to form. This idea of heat transfer is everything here. The vial is basically the bridge that allows thermal energy to move from the cold shelf of the freeze dryer through the glass and into the drug. If that bridge is thicker or thinner, it completely changes the freezing process and the quality of the final product. And ice nucleation, that's just a fancy term for the very first moment of freezing. Think of it like the first domino in a long chain. The physical design of the vial's base can actually tell that first domino where to fall, which sets up the entire frozen structure inside. It's amazing. All right, on to stage 2, primary drying. This is really the heart of the operation. It's where we start pulling all that frozen water out of the product. And here we go again. The packaging is running the show. How flat the bottom of the vial is, how well it makes contact with the shelf, directly affects how fast the drying happens. And the glass's ability to conduct heat ensures that the drug dries evenly into this consistent, stable structure that they call a cake. All of this is to make something called sublimation happen perfectly. It's this incredible physical process where you turn a solid ice directly into a gas, a vapor, completely skipping the liquid water phase. It's a really delicate dance, and the vial is leading the whole way. And that brings us to the final act, secondary drying. This is the finishing touch. It's where you remove those last really stubborn, hard to get rid of traces of moisture. This is what makes sure the drug is going to be stable for years on a shelf. But now the focus shifts. The vial has pretty much done its big job, and it hands the baton over to the stopper. The stopper's design and its material properties are what determine how much moisture is left in the end, and ultimately, that dictates the medicine's long-term stability.
You see, the stopper isn't just a dumb plug. For this phase, it has to act like a very specific kind of breathable membrane. It has to be just permeable enough to let those last little bits of water vapor escape. If it's too permeable, or not permeable enough, the whole multi-million-dollar batch could be ruined. So, when you put it all together, the freezing, the 2 drying stages, the vials, the stopper, a totally new picture starts to form. You realize you're not looking at a series of separate steps or individual components anymore. You're looking at one single, deeply interconnected system. And this right here is the absolute key takeaway. The most successful results, the best medicines, happen when separate jobs. They win when they treat it all as one integrated system, where every single part affects every other part. Which kind of leaves us with one last question to chew on. We've just seen how a simple glass vial is actually a piece of high-performance functional equipment. So it really makes you wonder, what other passive containers are out there in our world, quietly playing a much more active and much more important role than we've ever given them credit for?